Clinical trial data is the backbone of drug development. But before it can drive approvals, funding, or patient access, it must be clean, compliant, and insightful. SDTM standardization isn't just about regulations. It's about revealing truths hidden in the data. And Mushroom Solutions is here to automate that process. Manual SDTM validation creates delays, inconsistencies, and compliance risks. Every missing value, every formatting issue, puts your timeline, and your trial, at risk. But it doesn't have to be that way. Mushroom Solutions automates SDTM validation using AI-driven quality checks, real-time anomaly detection, and submission-ready reports. Our dashboard doesn't just show you the data, it reveals what matters most. This analysis presents insights from a Phase 3 clinical trial involving 1,000 patients. Using SDTM structured data, we explore key safety and efficacy metrics across different patient subgroups. The aim is to demonstrate how applying specific filters impacts clinical interpretation, helping trial teams make informed, targeted decisions. To illustrate how the dashboard responds to high-risk scenarios, we start by applying a filter for patients who experience severe adverse events. This filter allows us to observe how patient-level metrics change under more critical safety conditions. When the severe filter is applied, all dashboard visuals automatically adjust to reflect only those patients. We see that adverse event rates are highest in the high and medium dose groups. The response rate declines, indicating that patients with severe AEs tend to respond poorly to treatment. The treatment group proportion narrows as we isolate a smaller, high-risk subgroup. The age distribution shifts slightly toward older patients, and MELD scores increase. The MELD score, or Model for End-Stage Liver Disease, is a standard metric that estimates the severity of liver dysfunction. Higher MELD scores generally indicate poorer liver function and higher risk of complications. Here, increased MELD scores among severe AE patients confirm more advanced liver disease in this group. We also observe a rise in cardiac issues, suggesting compounded risk. This demonstrates how severe safety events can correlate with reduced efficacy and increased comorbidity, informing risk mitigation strategies during a trial. To further explore how clinical risk categories influence trial metrics, we apply a second filter, critical liver function status. This filter highlights patients with the most compromised hepatic function, a key consideration in trials involving liver endpoints. After applying the filter, we immediately see changes across the dashboard. The adverse event rate remains elevated, and response rates fall even further, indicating a clear impact of liver dysfunction on treatment effectiveness. The cardiac issue proportion rises again, and p-value sensitivity becomes a focal point. The p-value is a statistical measure that tells us whether the differences we observe, like changes in response rate, are likely due to actual effects or just random variation. A lower p-value, typically below 0.05, suggests that the result is statistically significant and unlikely to be due to chance. Here, the p-value confirms that the changes seen in this critical group are meaningful from a statistical standpoint. Higher MELD scores, more frequent encephalopathy, and worsening kidney function are evident, reflecting multi-organ strain in this group. The biomarker profile also shifts, helping researchers identify altered biological responses in these patients. This example shows how filtering by liver function status supports risk stratification and can influence inclusion or exclusion criteria. Lastly, to demonstrate how demographic subgroups influence clinical outcomes, we apply a filter for Asian ethnicity. This enables us to assess trial performance for a specific population. Once the filter is applied, all charts reflect patient metrics unique to the Asian subgroup. The gender distribution is balanced, but MELD scores by treatment group vary, revealing potential differences in baseline disease severity. Treatment effectiveness changes, giving us insights into how this subgroup responds to therapy. We also see new trends in adverse event patterns, kidney function, and risk factor prevalence, such as obesity, diabetes, and hypertension. This analysis helps support diversity in clinical trials and ensures that findings are representative and actionable across populations. SDTM data holds more than compliance, it holds insight. With Mushroom Solutions, uncover clinical insights through intelligent segmentation, AI, and automation to drive clinical decisions and regulatory success. Let's transform your trial outcomes. Visit our website or contact us directly. With Mushroom Solutions, make life better.